So let's have a little brief intro to nucleic acids. Nucleic acids you've absolutely heard of before. They're DNA and RNA, so deoxyribonucleic acid and ribonucleic acids. The only difference between DNA and RNA is the presence or absence of a particular OH group on the sugar. So for both nucleic acids, they can be split into three components. There's the phosphate group, which is just a phosphorus with a bunch of oxygens, and it has a negative charge to it overall. Then that is connected to a sugar. It's a ribose sugar or a deoxyribose sugar, which is just a pentose, so it has a little 5 ring to it. So we have the sugar portion. And then finally, there is what makes each nucleic acid special. So there's the nitrogen-containing base. So this overall structure is identical between both deoxyribonucleic acid and ribonucleic acid. The only difference is the presence of an OH for a ribo. So this one would be a ribose, a ribonucleic acid, and then this one down here would be a deoxy, where instead of having an OH, it just has a single hydrogen. Now, this difference is actually gigantic because, as you may remember if you watched the carbohydrate one, OH groups are very reactive. So H is reactive, and so if you have a no OH group here, this actually makes DNA pretty stable because once DNA has polymerized, it actually doesn't have this OH anymore except at the very end. So it's basically stripped away all of its OHs, and it doesn't have any OH here, so it just has a bunch of hydrogens. Whereas RNA actually will continue to have all of these OH groups along its entire chain length. So RNA covered with all of these very reactive groups. DNA is not, so it's extremely stable. Because RNA is unstable in the cell, or at least in eukaryotic cells, or in cells in general, it has temporary uses, as it can't be relied upon to still be there after a certain amount of time. So the RNAs that you see are things like tRNAs, ribosomal RNAs, messenger RNAs, microRNAs, small nuclear RNAs, a bunch of little pieces of nucleic acid information, but that are not supposed to be there forever. The only things that actually use RNA as a genome would be viruses. So viruses use RNA, some of them use RNA as a genome, and it's also what makes them so unstable and so prone to mutating. So you'll see that RNA genome viruses tend to mutate a lot faster than a DNA genome virus would. And viruses already mutate relatively fast given the speed of their copying. Just the overall nature of a nucleotide or nucleic acid, rather, is that you have this negative charge on the phosphate group for every single nucleotide. So you'll have the negative charge, you'll have the sugar, which is also polar, and then you'll have the nitrogen-containing base. So this whole thing is actually quite polar. It's not going to be wandering through any membranes. But then the negative phosphate actually drives a lot of things with DNA, just in terms of its overall structure, because technically as a chain, it's relatively flexible, but it can't actually bend in all ways. Polymers that have negative charge could only bend in certain directions, because if you were to bend this where you have your phosphate groups on the outside, so these would be our little phosphate groups all on the outside, that's great because the negative charges are all kept away from each other. However, if you were to bend with the bases facing outward and the phosphate groups facing inward, that would not be so good because the phosphate groups would be interacting with each other, and that is not so happiness-inducing. So you actually see that this limits the structures that DNA and RNA can take when they're single-stranded or even when they're making double strands, is that you'll always find it where you'll have the negative charged phosphate groups all pointing to the outside, and it will bend in ways that the phosphate groups never have to directly interact with each other. Instead, it's only the bases that have to directly interact with each other. Also, the fact that RNA and DNA have an overall negative charge is something that biologists have taken advantage of for decades, which is the fact that you can extract it using a positive charge. So you're actually able to isolate DNA and RNA relatively easily. It's why it's so easy to genotype things, because it's not that difficult to extract DNA from the soup. Finally, let's get into the nitrogenous base part itself. So this is what gives each nucleic acid the letter that it's assigned in a sequence. So the phosphate and sugar, if you're either talking about DNA or an RNA, that's always going to be the same, but the nitrogen-containing base is going to be different for each of the 4 DNA bases that you'll see. So you'll have adenine, guanine, cytosine, thymine, or AGCT, and uracil is something that you'd see in RNA, and it's just replacing the T. So the adenine and thymine base pair with each other, which is there's hydrogen bonding that actually can occur between opposing strands with adenine and thymine, and hydrogen bonding that occurs between opposing strands with guanine and cytosine. The way that these are set up, because remember, hydrogen bonding is a partial positive, interested in a partial negative, just the way that all of these sorts of intermolecular polar interactions occur. Because it's a partial positive and a partial negative, with the adenine and thymine, the partial positives and partial negatives are lined up where they're going to be happy with each other. But if you were to try to do the same with a guanine and a thymine, or a cytosine and an adenine, what you would end up getting is pluses and minuses, pluses and pluses interacting with each other instead of pluses and minuses. So you end up getting repulsion. So instead of having a plus lining up with a minus, you instead would get a plus lining up with another plus. And so that would be repellent. It's one of the things that makes it so that base pairing can actually work with specifically just adenine with thymine, and specifically just guanine with cytosine, and the other base pairing is not available because the hydrogens don't match up. So DNA as a polymer, you get a polymerization where you have phosphate sugar base. So you have along the backbone just a lot of phosphate groups for every single base, and a connecting sugar between all of them. This has a very particular order to it. So DNA, if you were to look at each strand, has what is referred to as the 5' end, where the phosphate is hanging off of the end, and at the opposite side it will have the 3' end, which is where the sugar is going to be hanging off. If you're to look at the opposing strand, you'd see that when you have the phosphate group sticking off of one, the other strand is going to have the sugar group sticking off, so they are anti-parallel, where you go 5' to 3', and then if you were to look at the opposing strand, it is going 3' to 5'. DNA itself is only polymerized in one direction. It is polymerized in the 5' to 3' direction because it happens by using that reactive free sugar. So you need to always have a free sugar with a reactive OH group in order to add to it. One of the cool things, of course, about DNA being set up this way, where you have this base pairing going on between each of the letters, that's very specific, where you can only get guanine base pairing with cytosine, only get adenine base pairing with thymine, is that you only need one strand to actually know the sequence of both of them. Once you have one strand, you'll know exactly how the matching strand must be lined up. And it's one of the ways that you can, that the cell is able to really have duplicate information for every single gene that it contains.